Start recording.
Introduce themselves and
Okay.
what we're going to do today.
All right. Well, I'll just say that obviously I'm Sam Droege, and Tom I've worked with and given him a bunch of specimens that have turned into some really interesting things that we might touch on here and there. And I'm sure that since that time he's added a lot more information about Epeolus, and I use his guide all the time. We are in the slow process of integrating a lot of his work into Discover Life. I think we've ingested all his pictures, but I'm going to turn it over to you, Tom. You can give a little bit of background, and then we'll just get into it. And you can run it how you want, and we'll back you up. But basically, just I think you've talked with Clare about just stepping through the guide. And these are technical people, so they're interested in interpretations of the characters and how you go about thinking about it, and then questions may pop up.
Okay. Well, thanks, Sam. So yes, I'm Tom Onuferko. I'm a taxonomist mainly, and I've revised the bee genus Epeolus for almost all New World species, and I've lately been working on taxonomic revisions of other bee genera, including Ceremolecta, which is now known as Brachymolecta, and I also have a few papers on the way revising groups of the bee genus Triepeolus. But today I'll be talking about Epeolus. It's a large genus, but mid-sized in North America. So around the world, there's more than 110 species. But in North America, north of Mexico, there are 44. That's the current number of valid species. And in the Americas, including Central America and South America, there's about 59 species in the genus. But I'm just going to focus on the ones occurring in the United States and Canada. There's no species that are endemic to Canada, so all the ones that occur in Canada also occur in the US. As Sam mentioned, the key on the Discover Life for Epeolus right now is outdated. I think it's mainly based on Mitchell's key, although the key that I will be going through today is eventually going to be posted and shared in a Discover Life friendly format with Discover Life terminology for those of you who are more comfortable with that. But for now, I'll just point you to my the key published in my Zuki's revision, and that link is available in the chat here. So yeah, I'm going to have to share my screen in a second.
It was also sent out in the email that comes out for people.
Okay. So let me just...
On the chat.
Okay. So does everyone see that?
Yes.
Okay. There's also a link to iNaturalist. So Epeolus, if you're not that familiar with the genus, they're rather photogenic bees, and a lot of people are imaging them. And they're also one of the few bees that can actually be identified from images of live specimens. Generally, what's very important in Epeolus identification are the patterns of pubescence on the mesosoma and metasoma, as well as integument coloration. There are other, more subtle differences in characters that separate species, but they are one of the rare groups of bees that can be identified on the wing. And I actually, I go onto iNaturalist a lot, and let me just pull this up.
I like Claire's comment before the talk. He said, yeah, Tom's on iNaturalist aggressively.
I don't know about aggressive, but I do check it regularly. But there's in addition to wanting to communicate taxon concepts to other people, I use it as a source of data. More lately in my research papers, I've been citing iNaturalist records under non-preserved material, because you get a lot of interesting data points on maps for species. So it's a very rich resource. And here I've filtered out all the Epeolus records on there. I'll limit it to the United States. Just so we don't get other species. And this is
Mhm.
a good resource because it also gives you an idea of how commonly encountered or observed various species are. So you can see that by far the most photographed specimens are Epeolus bifasciatus and Epeolus scutularis. That gives you an idea of what you might be more likely to encounter in the field in your own biodiversity surveys. So I'll refer to this periodically as I'm keying specimens or going through the key, just so you can get a sense of the variation of different features and how they look from different angles. But yeah, so that's the link to that is also in the chat. And I'll just go back to my revision. So there was one species that was discovered after this was published and named and described in 2022. So that's not included in this key. I've also, so the species is described in this paper in the journal Insectamundi. It's limited to largely to the Pacific Northwest. So probably won't be that commonly encountered, but it does include a modification to the Zuki's key that enables the identification of this species, and it has been incorporated into what will be the Discover Life key. So that will be complete when it's up. But just to let you know, that one species is missing from this particular paper. So I think separating Epeolus from Triepeolus has already been discussed in a previous recording, is my understanding. So I won't go through those details too much, but there is a generic diagnosis in here that you can consult if you're not entirely sure. If you have, here it is, if you have Epeolus or Triepeolus, but I will skip that. The terminology used in this publication largely follows Michener 2007, but there are some terms that I've adopted from Molly Reitmeyer's works, and they're in reference to the patterns of pubescence on the mesosome and metosome, and they're described here. I'll explain what they are in the key, but if you're ever not sure what I'm talking about, you can just refer back to this particular image.
Tom, just to interrupt very briefly, is it Epeolus? Is it Lectoides? Is that the one that looks a lot like a Triepeolus on the surface?
There are different species that may be confused with Triepeolus. I think it's so that Epeolus lectoides is an example of a species that has where the pseudopygidial area is more elongate and almost bell-shaped. There's quite a few species like that, and almost all are in the southeast. But that is probably the most commonly encountered one in that group. There's also, confusingly, a species of Triepeolus that has a very Epeolus-like pseudopygidial area. I think Triepeolus millenarius is the species that has, if you were to just look at the pseudopygidial area, you would think it's an Epeolus. But if you look at the protruding S6, it's very clearly a Triepeolus. There are, other features that might not have been covered that separate the two genera. One of the males, as most people know, are much more difficult to tell apart from females. But there are no species of Triepeolus that have an inner tooth on the mandible. In all species of Triepeolus, the mandibles are simple. And in most species of Epeolus, there's an inner tooth, but there are exceptions. So it's not 100% reliable. For separating males, I think probably the most reliable means of doing so is by looking at the genitalia, but that's difficult to do because they have to be excised, and they aren't necessarily excised in specimens once they've been pinned. I can briefly pull up Molly Reitmeyer's revision and show that. Let me
I think
just,
even though we have covered it, I think at the start here, this is good to just talk about that a little bit because people are come into the online recorded sessions in no particular order. So yeah, just a little bit because I know I've been tricked. Lectoides is the one that's has tripped me up more than once because I make assumptions about, the pseudopygidial area and things like that. And I get caught.
yeah. So there
Because
are,
I'll try and key it out in Molly's key, and it's like, this is really not coming out anywhere.
Yeah, my key doesn't work very well for Epeolus.
Right.
But yeah, and males are generally females are generally easier to tell apart, especially if you look at the S6, the S6 in the hidden sixth sternum, which has to be excised, but usually protrudes a little bit. It's diagnostic for Epeolus. There's no other Epeolini genera that have anything looking, remotely similar. But males are much more difficult. But one way to reliably do so, I don't know if you can see this. Can you see the screen with?
We can see your cursor. Mhm.
Okay. So this is an illustration of genital capsules and profile. Beg your pardon?
What'd you say, Claire?
You turn up your speaker a little bit.
Yes.
Sorry
Okay.
to interrupt.
So this is a picture of genital capsules and profile in Molly Reitmeyer's dissertation, published in 2006. So in Epeolus, the gonostylus has this really unique bulging lobe that is absent in Epeolus. This, I think, is the genitalia of Epeolus epeolurus, and this is I think, an African species of Epeolus. And there's also, if in these are images of the penis, and an Epeolus in most species, and there are some exceptions. The penis itself has a pair of these large fleshy lateral lobes. Those are absent in Epeolus, and they're also absent in Epeolus bifasciatus and the species that are in the Trophocleptria group. But those are, if you're really not sure and you don't want to, go through the trouble of barcoding specimens, or something like that, you can always excise the terminalia. I should also mention that I am happy to assist people with identifying specimens. Every once in a while, I'll get a request. Someone might send me images of specimens. They're unsure of the ID and they want me to check. I'm more than happy to do that. I don't, as I said, these requests aren't streaming in large numbers. This is not a genus like Lasioglossum, where I'm sure Jason gets tired of repeated requests to identify large numbers of specimens. These don't tend to be collected in large numbers in bee surveys if they show up at all. So I'm always happy to check IDs. And the only thing that I ask is that if this is for a paper and in the methodology, it would be good to cite the taxon concepts upon which the IDs were based. But yeah, I'm happy to do so and don't expect anything in return. So feel free to email me. And even today, if you have pictures that you want to upload at any point, I'm happy to take a look and I'll let you know what I think. So I'll jump right to the key. It's at the
Tom, just one observation that in terms of commonness, it seems to me that Epeolus might hold the record for the number of species that might be in a given area and the uncommonness of those species. Like it's a really, really uncommon species. We almost we'll run on a yearly basis. We may not get any Epeolus into the lab, for example.
And in your state, you might very well have 10 to 12 species. So that is a good point. Epeolus probably will take the cake on that one, on that record, but especially in the Southwest. But yeah, Epeolus is there. It's amazing how diverse they are, and yet how infrequently encountered they are. That probably has to do with the fact that they're very closely tied to a single host genus, unlike genera like Coelioxys or Nomata. Epeolus have just parasitize a single genus of bees, and that is Calides. And yeah, there's interesting stories about that. So there's a current paper that's in prep, wherein a species that I described in this Zuke's paper, Epeolus andrii, when I described it, it was known from only 2 specimens collected in the Jug Bay wetlands area. And it's since popped up in Oklahoma, which is like really far out, but I barcoded a specimen. The holotype of the species happened to have been barcoded, and the barcodes match. They confirm what I suspected based on morphology, and it was collected in an area where there was tons of cuscuta, and those cuscuta, or daughter flowers, are being visited by, I think it was Calides ciliatus. I'm not entirely sure, or Ciliatoides, one of those two. And so this, it might be a whole lot more common than we thought, but who's collecting bees off this very specific plant? Evidently not a whole lot of people.
But also the original site, the Jug Bay site, that is also where they have collected Calides ciliatus, and is the only place in the state that we currently know about it. But again, who looks at daughter?
Yeah.
This was specifically, I would say, it's probably it seems like in both those cases that this is a wetland daughter species. It's difficult to tell the daughters apart too, but anyway, people should be looking in late fall, September, October, on daughter for all of the above.
And September is when I think all the specimens that I've seen have been collected. That reminds me of another point. So for each of these species, there is a range map. But take it as a grain of salt for the species that are represented by very few specimens. So this is an example of, that's where Epeolus andrii, that's the type locality, and since it's popped up in Oklahoma, for species that are far more abundant than this one, you might get an idea, like something Epeolus bifasciatus. I mean, that's a very distinctive species, but if you were to, if you thought you might have this species and it was really out of range, then, this is a pretty good, I think, pretty accurate representation of where you're most likely to, where you should find it. So the maps are useful. There are certain things that, like, Epeolus asperatus, I wouldn't expect to find far beyond California, given that all the records that I've seen are from that state. So if you have one in Kansas or Nebraska, that's probably another species that's very similar to it, probably Epeolus americana. So you can use the maps to help, to guide you in identifying things. I think that's one thing that's often missing from a lot of taxonomic papers is actual, distribution maps of species, but I do find that they're a useful tool. And I often check Discover Life records when I want to, verify that I have something and, determine whether it's plausible that what I have actually, or that my idea is plausible based on where I've collected the specimen. But anyways, I'll jump right to the key. So this key is fully illustrated. And one thing I like about this journal is that the couplets are all, so the figures, citations are all hyperlinked. So if you just go to the couplet and you click on one figure, it'll pop up right next to the couplet where you are. So there's no scrolling back and forth in this key, which is really nice. I structured this key to try to make it as evenly bifurcated as possible. So minimizing the number of steps required to identify all specimens to species. One of the issues with dichotomous keys is that often they're constructed in a way that a species is sort of crossed off one by one, and you may need to get to, through 40 couplets to identify the most common species in a genus. So I wanted to minimize, the amount of steps required to identify all species. And so that's why there might be, in the initial splits, there might be fewer characters to look at. ideally, you'd want to have as many features as possible to look at. But in this case, in the first couplet, I selected a character that is quite reliable, but it's just a single character, so you really have to get the hang of it. So the first character here refers to the posterior extent of the axillae, and this is relative to the length of the mesoscutellum. So I'll just blow up this picture here. And so these are, the axillae of an Epeolus species. This is Epeolus bifasciatus. Here's the mesoscutellum, and I've drawn a line of how far back it goes. And so the first half of the couplet basically leads to the species where the axillae extend further than 2/3 the length of the mesoscutellum. The other one is less than that. It's, might seem like an arbitrary cutoff, but it is quite reliable. There's only one species I found that has to be keyed in both places. That's Epeolus lectus. And so, regardless of which of those steps you take, you can still get to that species. But for the other ones, it's quite invariable. And so here I show an example of what the minimum posterior extent, and then you can look at examples of species where it goes even further than that. Here, like in Epeolus scutularis, it's pretty much at the posterior extent of the mesoscutellum. And then I'll just close these windows. And then if you look at the other half of the couplet, here's what the maximum posterior extent looks like. So it's pretty close to, it's only a little, they only slightly extend, not as far anteriorly as in the other couplet. This species is very rare. I don't think I've seen specimens of it collected more recently than the 1930s, so it may very well be extinct. This is the Epeolus banksii. That's a good one to keep your eyes open for. But there are examples of, axillae that are clearly much shorter than that. So in this species, Epeolus asperatus, it doesn't even extend past the mid-length of the scutellum. So when using this key, it really helps to have a calibrated, well, actually, it doesn't even need to be calibrated, an ocular micrometer within the eyepiece to measure. As you get more comfortable gauging the length, then it probably won't be necessary to use it. And it probably only really is necessary for the species where it's closest to the cutoff line. But generally, you can tell quite easily, where the axillae end up. And so, yeah, I'm sort of, Sam, do you want to pull up maybe an example of one of the specimens you have? I don't know if it makes sense.
Yeah.
To just try to, it can, doesn't matter which one. I think most of yours will key to the, will key out earlier in the key.
Yeah, I'll pull a scutularis since I have a lot of
Okay.
those. And while I'm setting it up, you can talk about other
Sure.
things because it'll take me a second to get it in the viewfinder and all that kind of good stuff.
So let me see if scutularis was the
It's the most common in Maryland.
it's a very
Which is insane.
common species. It's comes out late summer. I think you'll start encountering males in late July and August, and then through September here, at least in our area. But it is a very commonly encountered species of Epeolus. Like I've got 2 unit trays of it. In my own collection, there's 137 observations on iNaturalist. It's presumed host is Calides simulans, which is a common species on visits a variety of flowers, but I found it very most commonly associated with solidago, various goldenrod, but it does go to all sorts of other asters. So that is, one of the more common Epeolus species, especially in eastern North America. They get rarer out west. So that is a species in which the axilla go back almost all the way to the end of the scutellum, and quite obviously so. And so we'll skip ahead to couplet 2.
Well, if you want,
Or do you
I've got it on deck now. So we
Okay.
can, do you want me to flip it over?
Sure.
Okay.
Yeah.
So we'll go to screen and is it up? Do you see something?
Yes.
All right. Tom, you see it too?
Yep.
All right.
Yeah. So
axilla there.
yeah,
And
it's
your pointer small.
Oh, I'll make my pointer really big. So hang on for 1 second.
so I would, when looking at it, I would try to look at it kind of like it is in the image, just flat from, as from the top, as opposed to sort of in an oblique view, like as close to, I guess, what you'd consider a perfect dorsal view as possible. Because obviously, if you rotate the specimen, in some views it will look like the axillae don't go that far back, and in some it will. And so that is, one thing to consider is like match it up to how it's shown in the image when you're using the key. But in this species, they're so big that you can kind of see even where, that it goes past. There's sort of a ridge on the horizontal dorsal portion of the scutellum. Yeah, exactly where Sam's pointing has his cursor. You can see that even in that view, they extend past it. And the scutellum extends further down, but I think most of the comparisons that I make with reference to the axillae are just to the dorsal portion. And there's usually a ridge or carina at the end of it.
It's difficult to see because of lack of shadow here that there is a pretty strong edge along this line that I'm showing.
Yeah. So now I'll proceed to the next couplet. I don't know if
you can hit sharing. I'm stopping sharing, but I don't know if that flips to you.
It does not.
Yeah, so you'll have to share upper right.
Here we go. Do you see it now?
It looks like it's trying to load. There we go. Yep.
Okay. So we skipped ahead to second couplet because we can see how far back the axillae go. Here I basically just cross out a very easily identified species in North America, Epeolus bifasciatus, which has a whole bunch of unique features. Epeolus scutellaris doesn't have that. What those features are, I'll just compare. So Epeolus bifasciatus is unique among North American species because it's got these frontal protrusions. It's only members of this predominantly neotropical species group that have these protrusions on the frontal area. That group is referred to as the Trophocleptria group. It's not present in Epeolus scutellaris. Also, as the name indicates, bifasciatus has only 2 bands on the metazoma. This is because this species is it's a eumenine mimic. It's a potter wasp mimic. So if you've seen enough potter wasps, you'll notice that the second tergum is like really expanded. And this species gives the illusion of an expanded second tergum by having only a basal fascia on T1 and an apical fascia on T2. And so this you might not even immediately recognize as an Epeolus. You might recognize this as like a Uodinaris or something. So it's really quite distinctive. And so Epeolus scutellaris doesn't have that. It has bands on all the metasomal terga, well-developed apical fasciae on T1 through T4 in females or through T7 in in males. And I think those are the only characters there. So I don't know, Sam, if you want to pull the
Bifasciatus.
specimen back.
Oh, you want Scutularis back?
Yeah, just to show that
okay.
it doesn't have those features. Sorry. maybe it's better if you just hold, keep it next to you.
Keep it on deck.
Yeah.
Are we sorry, are your guides male and female or
That's
both?
an excellent point. So I didn't mention that at the start. Epeolus are largely sexually monomorphic.
Yeah.
So this particular key will work for both sexes. If there are any sex-specific features, then they're explicitly indicated as such in the key, and there are always features that can tell the other sex apart in that same couplet. So I think when we put when we started to put together the Discover Life equivalent, it will be separated by there will be female and male versions of the key. They'll largely be the same. But for those who are more used to, keying, using sex-specific keys, they'll have that available in that format. But this key that I'm going through today will work for both sexes. So here, as you can see, there's, distinct bands on all the metasomal trigger 1, 2, 3, 4, 5, 6. Oh, and a tiny little 7th band, I think, there too. But yeah, clearly more than in bifasciatus. And if you were to look closely at the head, you'd see that there's no clear protrusions.
We don't necessarily need to see that because it's so
it's
distinct.
showing, it's always seeing the absence, highlighting the absence of a feature isn't always necessarily useful. So I guess we can skip ahead.
Yeah. Okay.
Maybe, I don't know if I should just maybe I should read the couplets while you hold the specimen. Would that be easier, Sam, do you think? Or, okay.
I mean, so the way to go here is we could flip back and forth, and Claire can also, sometimes she'll hold the guide in place.
Okay.
And you could have pictures ready to go, and I could have a specimen if we want to kind of see in a live action shot. But so you're basically the boss, so you can tell me, like, I want to see Scutularis in this certain view or whatever.
Okay. So you're still sharing your screen, I see, right?
Yeah. So you just grab it when you want to go to look at
maybe we'll just do it that way, just so people can follow along, see where I am in the key too, as opposed to just
I think that's important.
Talking. So let me go back to the feature. So now, so we've ruled out Epeolus bifasciatus. So now we'll continue to couplet 3. This is a bit of a tricky one. It basically has ifs and if-then characters in it. But basically it separates the species that have enlarged axilli that aren't distinctly hooked on the inner margin and that are like dilated laterally, or arcuate is the term I think I use here. So basically it separates the species where the inner margin of the axilli are relatively straight and the outer margins are distinctly dilated. Does that make Sam? Are you seeing my cursor and everything? Is that?
Yeah. No, you're good.
Yeah.
So it's actually, when you talk about dilated, you might want to explain that a little bit more. What is dilated? Is it the border is dilated, or are you talking about its thickness, which we can't see?
So it's just the shape of the axilla. It's convex on the outer margin, as opposed to straight or relatively straight. So here, if you were to draw a line straight down, you can see that it's very clearly convex. So that's kind of what that's what I meant by dilated. Convex is probably a better term. And the inner margin of the axilla is relatively straight. The alternative, in the preceding couplet, there's a bunch of options. So one refers to how how far the free how elongated the free portion is.
Okay, I actually had lunch before I got here.
Oh, hold on.
I'm with you one little bit.
Oh, okay.
And so
Thank you.
Got it.
So I'll just read it out loud. So
Mhm.
is the free portion of the axilla approximately equal to 2/5 of its entire medial length or more, or is it less than that amount? So here you have to take a ruler to the screen, and that's what this red line indicates here. I drew a line where it begins to diverge from the scutellum, and how long is that free portion relative to the rest of the inner length of the axilla? Here you can see it's clearly shorter than 2/5. It's probably about 1/3 the length. But again, this is where an ocular micrometer comes in handy if you're not entirely sure. If you can't gauge, just looking at it. The other feature was,
So Tom, you talked about the medial length. Okay,
yeah,
so
the inner length could be another way to describe it. But yeah.
Okay, because medial, like I would have taken the center line of the. Anyway, that's good that you explained it.
Yeah, in this case, what I meant the margin that's closer to the middle, because there's I guess 3 margins to the axilla. The outer, but we can use the terms outer and inner if that's more intuitive. So so that's one option if the free portion is longer, then you would go to 4. If it's less than that, you potentially go to 11, but it has to be these characters in combination to go to that couplet. Because if you're looking at something that might be borderline, then look at whether the axilla is relatively straight on the outer margin. In this case, in Epeolus scutularis, it clearly isn't. It's clearly convex. Now, if you're still confused, then you can also look at the inner margin and see if there's a distinct hook. So is it distinctly concave on the is the free portion distinctly concave along the inner margin? Here it's relatively straight throughout, so it doesn't match the features in the first half of the couplet. In this case, we can see that it's clearly shorter than it was described in the first half, and that the latter margin is distinctly convex. So we proceed to the next one. Maybe I'll just show you a few examples of what the alternate states look like, just so you have an idea. So so here is an example of a species that is common in the Great Plains. You can see that the free portion of the axilla, I think it almost is like half the length of the axilla. You can also see that even though in this species the outer margin is convex, the inner margin is very much concave, so it looks like a hook. It's not, it's not completely straight throughout. Does that make sense to you, Sam?
Yeah, totally.
Okay. Let me just see what some of the other pictures had. So yeah, so here's an example of a species in which the free portion of the axilla are quite long, but the lateral margin is relatively straight. It's not distinctly convex, maybe only slightly convex, but clearly not as it is in Epeolus scutularis.
You can see, so some of the ambiguity, like this is always true for all these taxonomic keys. So I might look at that and say, well, the tip is a little bit hooked. You know what I mean? So, but the side is straight. So
yeah,
this
but no,
is
that's
why
okay because it's supposed to be
multiple
somewhat
pathways.
hooked.
Yeah,
it's supposed to be somewhat hooked in this species. That's
Mhm.
fine. It doesn't key to the same place as scutularis. It's in the first half of the couplet. The second half was it has to be those 2 features in combination, or
Yeah.
3 features in combination. Here it could be 1 or more of these features, but the fact that this outer margin is straight, that to me seems like the most obvious character that rules out the second half of the couplet. So I'll I think Yeah, so that one I wasn't a huge fan of this character when drafting it, but it is reliable if you get the hang of it. But I know that if then characters can be frustrating. I don't think I have too many of those in this key, but that is one that is unavoidable. So because we've picked this couplet, we'll skip ahead to this half of the couplet. We'll skip ahead to 11, so that it's none of these species. And now we'll look at the fasciae. So this one's a bit, this one's can be a little tougher because every once in a while you might get aberrant specimens, but it refers to whether or not the fasciae are interrupted medially. And Epeolus scutellars, they often are very narrowly on the first two terga, but they shouldn't be at all on T3 or any of the subsequent terga. So the species where they're medially interrupted, I think, are almost all southeastern, if I'm not mistaken. Yeah. So 4 species. This is the very rare Epeolus andrii that seems to be associated with Cuscuta. Unlike in the Epeolus scutellars that Sam pulled up, you can see that the terga are very clearly interrupted on the or the fasciae on the are very clearly interrupted on the first 3 terga and narrowed on the 4th, but the couplet refers just to these 3 and quite distinctly interrupted in the first tergum. And if you recall,
you can
Sam's
also yeah, you can also see, actually Ben Hollister was one who collected it, but the you can also see on that particular slide, you'll see this quite commonly, is that those white oppressed hairs often disappear if there's even a little bit of goop on the specimen. And so sometimes you have to really look carefully to see patterns in hair bands if you have a goopy, let's call it a goopy specimen, because you can see like those marks there where they've just gone. I think the hairs may be removed, but I think they're also quite likely they could be simply covered with some junk.
I think in that
They turn
case,
black.
it looks like they're removed because I can see the pits underneath in some of
Okay.
the, like on T2, but that is a very good point. So this key assumes that your specimens are more or less in optimal condition. And if you have a specimen wherein the pubescence is badly stained or rubbed off, and sometimes there's no helping it, sometimes just the specimen that you collect in the field is, towards the end of its flight season. And, in this case, you can see the wings are a little tattered.
Yep.
So there might simply be no workaround, unfortunately. But yeah, that is a good thing to keep in mind. I have found that if the pubescence is not rubbed off but stained, sometimes resoaking a specimen in like 70% ethanol for a day and then repinning it can restore the coloration to almost what it was originally. It's I've had success doing that. If there's no other way that you can identify a specimen because like the terga look black, you might want to consider doing that. And for some reason, sometimes you might have to experiment. If ethanol doesn't work, maybe just rather than soaking the entire specimen, you can take a little piece of tissue paper and wet it with water and just put it on the metasoma, let it sit there. And if the tissue paper starts becoming like, yellow, then it's starting to pull up all sorts of gunk that from the surface, and that may help restore the color. And sometimes I found that, maybe just as close to pure ethanol as possible will work, where 70% ethanol or water doesn't. So there might be some experimentation involved, but that is an important thing to consider because, as I mentioned, a lot of the identification of Epeolus is based on the patterns of pubescence on the mesosoma and metasoma. Whereas for Triepeolus, like the pseudopygidial area is immensely important in coming to species level IDs. For Epeolus, I would say, it's the axillae and the patterns of pubescence. Those two things are really important. And so that's something to keep in mind too when you're, when you're pinning things, make sure that you're not pulling things straight out of pan traps and sticking a pin through, because that can tarnish the or badly stain the pubescence. So I'll just pull up some other.
Acetone. Acetone always works too.
So
Yeah.
you said something about pitting underneath those bands. Did I catch that right? What was that about?
Sorry, could you repeat that?
Did you say something about there's pitting or not pitting underneath the bands that might give you a clue? Or did I mishear that?
Oh, pitting. Okay, like the yes. So no, I don't. The yeah. So actually, well, there. That's true. Actually, no. Now that I think of it, so the discs of these turga, they're often generally covered in pubescence. It's just a darker pubescence, and it's sparser. So if you see that there's an area that's completely devoid of pubescence, that's an indication that pubescence was wiped off. So it's hard to see in this picture, but you can kind of see the specimen at one point was in solution because the hairs of, the small CT have kind of clumped up. And if you look along, on the disc of the metasoma, you can see there's sort of like, short lines here. Those are the hairs. And here, where the fascia used to be, you can see it's just pitted, but there's nothing else. So that would be an indication that the hairs are wiped off. Does that answer your question?
I think so.
I think if I may interpret there too, is pitting ever useful in identification?
There are species that have sparser pits on the metasoma. It's actually Lectoides and Glabratus are two species where the pits are quite sparse and also very fine. And then there's a species Lectus, which is closely related to those two, where the pits are very coarse and quite dense, and that feature is important. And Lectus is very similar to Lectoides, but that is the feature that essentially one of the main features that sets those species apart. So yeah, there are some species for which the pits are a useful character, but for most, the pit density is, it's generally, approximately the interspaces are equal to a pit diameter for most species in the genus. Okay, so let me just see what other species are in that. Yeah, so here's another species. What was that one? It didn't open up. Oh, here we go. Epeolus deripi. This is the species that's not in Discover Life because it wasn't described until I revised the genus. It was missed by Mitchell, and Brumley didn't look at the eastern species, but it was unknown to science. It's found only in peninsular Florida and coastal Georgia, but here you can see like the bands are very clearly interrupted immediately. They're even reduced, on the subsequent turga. So that matches the first half of this couplet. And then Floridensis is another one where the bands are very distinctly interrupted immediately on the first, I think, 3 or 4 turga. Oops, sorry. Just close those. So we'll skip ahead to the ones where it's not, where the bands aren't interrupted immediately on those first 3 turgas. So they're complete or only extremely narrowly interrupted. So what are some of the species in this key? So Epeolus autumnalis is an example where they're, here in T3 is complete, T2 is very narrowly interrupted, T1 is more distinctly interrupted immediately.
So Tom,
Let's go.
talk about interrupted and narrowly interrupted. So when you talk about narrowly, is there a cutoff between interrupted and narrowly interrupted? Or is it like it when you see it?
Yeah, I think the more important thing is, I think, what it's like on T3. T3 has to be complete. There's no, it's somewhat, I don't want to say subjective. There's no strict cutoff here. The ones where it's I would say distinctly interrupted, it's almost like they're separated into some rounded lobes medially, as opposed to just sort of tapering to and almost connecting. But I think the more important feature in this couplet is what is it like on T3? T3 or T sorry. Yeah, and it's in the species, the key here, if it's interrupted, it's almost it'll be on T1 and T2, maybe very rarely on T3. But this is the more, I think Epeolus autumnalis might have been a bad example, because that's kind of a species where it's a little more borderline, whereas here in Epeolus scutularis, it clearly isn't. That little line here is pubescence that was scratched off, so that's another thing to be mindful of. It looks like the middle point is actually here, but in these bands, like here they're touching, here they're narrowed, complete, and this should be complete, if that makes
We see that in Nomata all the time too, where you can tell that they might not necessarily have a complete band because there's a faded area, a necked down area, or some subtlety.
Yeah, I've never known how to interpret that, because it looks like it's a continuous band, but the middle part is just a very dark color. And so I was never sure if Mitchell referred to that as complete or interrupted. But that is in this case, the color, the bands are made entirely of they're made of pubescence. There's no integument coloration doesn't play a part. And you do have to be mindful of the fact that it might be rubbed off medially just where you need to look at it. So just because that's an area where you have to look and look at the pits to see is the area where there's no bands, is it entirely glabrous, is it entirely smooth? Because I have collected Epeolus scutularis in the field where the middle portion was just rubbed out.
Right. So another point that when you are comparing, say, maculations or color patterns in general, and you're comparing them to pubescent patterns, the pubescence patterns are much more stable within a species. In other words, there is variation, but not nearly as much as, like, maybe talk a little bit about, so like in scutularis, there's often a lot of red, but my impression would be that you can go bright red to darker. And so is there
and in males it's often the scutellum, the mesoscutellum is often entirely black, and much of the axillae are as well. So it's so it's not just even the color, but the extent to which those areas are covered by I use the jargony term ferruginous throughout the throughout the paper, but I'm basically referring to rust colored, anything from orange to to brownish integument coloration, different from this dark brown or almost black coloration, I guess. Ferruginous versus pisceus, or you can call it reddish brown versus dark brown black. But even there is variability, and even among species as well. So like in glabratus, it's much more of a deep blood red, almost like reminiscent to like Spicotti's coloration. And that's common to a lot of Hymenoptera from Florida for whatever reason. Species from Florida often have very dark red integument coloration, and here it's like a different kind of red. But I have tested these characters with many specimens, and have tested the key with other not experts on the genus, but other melitologists to get an idea of what works and doesn't. So it has been fine-tuned. but you may very well encounter aberrant specimens every once in a while, that for what or damaged specimens for which the key might not work so well. But so based on the fact that in Scutularis and in the specimen that Sam showed before, that the facie are they're not distinctly separated immediately into and I haven't gone through and in the preceding couplet, the axilla and scutellum or mesoscutellum are both ferruginous. Here the coloration may be variable. This will include species where the two are black. So if we go back to autumnalis, if you weren't sure how to score this because these two bands are medially interrupted and this one is narrowed, but it's touching, but you're still unsure. The fact that the axilla and scutellum are entirely black in this species tells you that it'll key out in this half of the couplet. So look at all the characters in the couplets when making a decision, because sometimes it's features in combination that will determine where you have to go. So in this case, it's not. We'll skip ahead to 16.
I'm actually gonna give us a time check. We are at.
Oh wow!
We have
Okay.
time.
2, so this is a good time to pause. And I can take notes.
Time flies.
For picking up next time.
God, has it been an
So is
hour
this
already? Okay.
Yeah. Is this a good stopping place here then? I feel like
Okay.
we've tackled some of the really core basic, some of these measurement things and talk about how the patterns are portrayed and that. So that's solid. And then we're going to get
Haven't
into
even keyed one
probably
species.
more than nuance. Yeah. We could do bifasciatus now.
Yeah, I should have done that one. That was only two steps, or I think it was 2 couplets.
No,
Yeah.
it's
So,
solid.
Oh,
so do we want to pick are we gonna pick up then next week? I don't know how you wanna.
I think Claire and I talked about this. There's been different approaches, but I think, in terms of like people tracking from one talk to the next, that literally just going through the key is really useful and helpful
Okay.
for wrapping their minds around these kinds of
Okay.
things. There can be times where there can be some hand waving, like you have to look at this group a little more detailed or whatever. But I think that's what these set of talks are about, is that we get to go into the detail where and learn from you about what were you thinking about when you were seeing these different things? How do you approach the identification and the use of these characters so that people can carry on when they're struggling. They can look this back up. It's all archived.
Okay.
So this
So
is good.
yeah, I'll leave it to you guys then to decide whether you want to maybe do a subsequent session, but just letting you know that if you did decide, yeah.
Oh, we are.
I'd be okay.
Yeah. We're just
Okay.
assuming that, Tom.
Okay.
It
So I
might be
am
another
confirming my
2
availability
years.
next Wednesday if you want to do it then, but we can discuss via
Yeah.
email.
Okay.
So maybe at this point, did anyone have any questions in the chat?
What do you?
No, but it is open for question for sure.
Yeah. It turns out most of the time people don't have too many questions,
Yeah.
because it's when you start getting down to some of the things that they have struggled with that they're going to start asking questions like Pusillus versus Scutularis or whatever. And then people are encouraged to bring those questions and to, bring some pictures and things too. And then that's useful when you're struggling to look at something tricky.
Yeah. And Pusillus is one of the, Pusillus versus Scutularis is one that can be difficult to tell apart.
I've spent a bit of time. I'm looking for that to myself. But
the males are really easy. That's the if the pleura are entirely covered by pale pubescence, then for sure it's Pusillus or in that group. But for Eastern North America, there's only Pusillus in the Pusillus group. But yeah, just I don't think I see any questions in the chat. Okay. Yeah, so if there's no,
I was looking around on our Discover Life pictures, so
Uh-huh.
you talked about iNet. Definitely we'll advertise that some more. There's a link in the chat. But if they want in situ photos, your photos are on Discover Life. You search the thumbnails sometimes, but there's also others if you're looking for different angles.
yeah.
That's all.
Also, I'll add to iNat, you can see exactly who made the identifications if you look at the, if you click on an observation. And so if you're trying to decide whether or not the ID is trustworthy, you can see if my name's on there. Then you're going by my call, so take that as what you make.
Right.
But sometimes, there's iNaturalist often suggests I think it suggests IDs when you upload things and you're not sure what it is.
Right.
Yeah, and I think by default it seems to suggest the most common species in a genus. So like there's almost any Epeolus that you might put up will be suggested as Epeolus lunatus. And I think probably Scutularis will be an Epeolus that might be. So take a look at who actually did the IDs, not necessarily just if it's needs ID or research grade. If it says it needs ID, but that's because I'm the only person that added an ID and no one has corroborated it, that would be more trustworthy than 5 people who don't know the genus having added their identifications. But it is, I find it's a very useful tool. If you're trying to get, I think one of the most difficult things when identifying any group is having reference material. And Epeolus is one of those genera that's not so commonly encountered. So getting access to a good reference collection might be very difficult. And so that's where finding images of specimens really helps. Discover Life is obviously a great resource for that. But iNaturalist and to some extent Bug Guide are pretty useful too.
Yeah. Well, thank you very much, Tom. We appreciate your time. We've been angling to have you on here for a while, and here you are. And we'll set the hook and see if we can get you back next week or whatever the schedule is that permits. Several weeks. Claire's,
Okay, well, thanks.
All right.
Thanks, Sam. Thanks everyone for attending. I see some names that I recognize. So glad you're all tuned in and look forward to the next session.
Okay.
Cheers.
Thanks, Tom.